So chapter 1 is kind of an overview of everything that we're going to talk about this semester. Some of it should be review. You guys should have had it in your regular biology classes. I know some of you were in my regular biology class, so I know some of you had this. But your notes have blanks throughout the presentation. So as I go through them, we're going to fill in the blanks. So I know you guys didn't have a chance to write these down, which is fine today. I'm going to post this recording, so if I go too fast or you miss something, you guys can always go back and watch it. So, question. You guys don't have to write this one down. This is just my question to you. What diseases and disorders can you inherit from your parents or grandparents? So you can either unmute yourself or type it in the chat box. I like these kind of notes. Good, Shay. I'm glad. So what do you think? What can you inherit from your parents or grandparents?
I know that if you get older, you can inherit Alzheimer's.
Alzheimer's? Yeah. Good.
But Miss Bruce, could be like also like a mental disease and like not mental disease, but like
a mental disorder?
Disorder.
Yeah, mental ones are called disorders.
Yeah,
like
mental
what?
disorder, like for example, like what were we talking about? Down syndrome, syndrome of Down.
So Down syndrome is not hereditary.
Is that
that's chromosomal. We're going to talk about that.
Okay.
That actually has to do with the woman. That actually has to do with the woman. Alzheimer's.
Yeah.
Adrian, we stole your answer. Diabetes is a good one. Diabetes can be hereditary. Good. Anything else? Any other ailments you can think? Any other disorders or sicknesses that your parents or grandparents suffer from that you think could be hereditary?
Isn't schizophrenia hereditary?
It could be. So schizophrenia is a little bit of both, a little bit environmental, a little bit hereditary. Diabetes actually can be too, depending on if it's type 1 or type 2. Blood pressure is one. Both of my parents have high blood pressure, so therefore I have high blood pressure. And then certain cancers are also hereditary. Good. All right. So translational medicine is the field where you move laboratory research from the lab into tools that doctors can use to diagnose and actually treat diseases and disorders. So like I said, in this slide that you guys have, you have blanks. So you can either write in your blanks if you need to write down the notes. Writing down the notes is always good. It always helps you remember better. If you don't have time, you can make a copy of that presentation with the blanks. Go to File, make a copy, and you can just type in the blanks. Immunotherapy is another field. It treats diseases by stimulating the immune system to recognize and attack abnormal cells. This is what vaccines are. Vaccines stimulate our immune system to fight different diseases and disorders. And then it has been used to treat different forms of leukemia by encouraging an immune response against cancerous cells. And then I'll usually wait like 10 seconds, let you guys catch up, and then I'm going to move on to the next one. So understanding genetics is essential to understanding life. These next few slides should be a review for you guys. You guys should have heard all of these words and all of these concepts before. So genetics is a scientific study of heredity, how traits are passed on from one generation to the next. It includes a study of how genes function in cells. And we talked about this if you're in my regular biology class. We talked about this if you're in my Bio134 class. We talked about genetics. Gene is the fundamental unit of heredity. It's the structural and functional unit of genetics, composed of a unique DNA sequence. DNA consists of A, T, G, and C, our 4 nucleotides. Does anyone remember how they are put together? They're put together in a certain way. They don't all pair together. Remember which one goes with which one?
Isn't it adenine goes with thymine and cytosine goes with guanine?
Yep. So I like to remember it, A and T, the letters are like straight. So the straight letters go together. And then G and C are the curly letters. So G and C always stick together. DNA sequence, the combination or sequence of the 4 nucleotides in a DNA molecules. This is what stores the genetic information of an individual, and it defines which amino acids will be used to make specific proteins in the cell. So you each have a specific unique DNA sequence that makes your proteins be expressed a certain way. Some people have the protein that gets expressed for brown eyes. Some people have the protein that gets expressed for blue eyes. And it just depends on the order of the nucleotides. Is it ACTG or is it GTCA? Or is it CGTA? Everybody's is different. All right, amino acids make up polypeptide chains, which become functional proteins when folded into specific 3-dimensional shapes. So protein, they fold this way and they keep folding. They make little shapes like this. And then eventually amino acids get together and they're a huge structure. So this is just an overview. We're going to get into more details in our chapters as we go along. The foundation of genetics were established in the 19th century by Gregor Mendel. He's called the father of genetics, and you guys have an assignment on him, a little 5-10 minute ed puzzle that you're going to watch and answer questions about him and his work. Mendel's experiments showed that traits are passed from parents to offspring. His conclusions: each parent carries 2 genes that control a specific trait. Each parent contributes 1 copy from each gene pair to each offspring. So we all have 2 pairs of every single gene that we have in our DNA. But when we have kids, we're only going to pass on 1 out of those 2. Pairs separate from each other during the formation of egg and sperm, meiosis. And then when egg and sperm fuse during fertilization, genes from the mother and father become a new gene pair. Genes are contained on chromosomes, which are located in the nucleus of a eukaryotic cell. So here's the entire cell on the left. You get a little closer, you can see the nucleus. The little X structures are called chromosomes. As you get closer to chromosomes, they have a banding pattern, which are all these stripes. You get closer to chromosomes, you can see the DNA. And then a gene is a segment of DNA on the big long chain. There's 4 approaches to studying genes. So how do scientists study genes? 1, they study transmission genetics. A pedigree analysis is kind of like a family tree, but they just concentrate on 1 specific trait that moves through that family tree. There's a field called cytogenics, in which they use a karyotype to make sure people have the right amount of chromosomes. You're only supposed to have a certain number if you have more or less. I know that if you were in my class, we put together karyotypes. There's molecular genetics, recombinant DNA technology. That's all the fancy stuff. You can produce millions of copies of desired genes or clones for study. You can use it for gene therapy, like inserting the gene for insulin into a diabetic so they can make their own insulin. The Human Genome Project and genomics is born out of this field. And then there's population genetics is the 4th approach. Pedigree analysis. So this is what the family tree looks like on a pedigree. It's a construction of family trees used to follow transmission or inheritance of genetic traits in families. Usually it follows things like diseases, although you can do simple ones with traits like eye color. The circles and the squares are different members of the family. Circles are always females, squares are always males. And if they're colored in, like the black ones, that means they have the affected trait, whatever trait you're studying. But we'll get into this a little more and how to read pedigrees. Karyotyping. A karyotype is a complete set of chromosomes from a dividing cell. It's photographed. Each chromosome is paired and arranged by size and shape in a standard order. You got 22 pairs of regular chromosomes. And then right here, the X and the Y determines your gender. Does anyone know what this is? Who has an X and a Y? What gender?
Are for females and Y's are males. So if you have an X and a Y, you're a male.
Very good. I like to remember the word boy ends in Y. So if you see a Y, it's a boy. Biotechnology using recombinant DNA has developed genetically modified crops. Almost all of our food is genetically modified. I know people always talk about, oh, genetically modified foods, non-GMO foods. All of our food has been genetically modified over the last thousands of years. So when we get into this chapter, we'll get into GMO crops a little more in food. They always want to insert a gene that will make it last longer or will make it resistant to certain kind of pests or pesticides. The impact of genetics on social policy and law. This is actually going to be our weekly discussion post. Eugenics is the attempt to improve the human species by selective breeding, which by itself, just by definition, okay, that sounds okay, the attempt to improve the human species by selective breeding. However, there are more downsides than there are upsides. So it is a flawed theory based on hereditary traits only, ignoring environmental effects. There are environmental effects to our health. In the United States, there were laws restricting reproductive rights, required certain individuals to be sterilized. You can look up forced sterilization. It's been happening since the beginning of our country, and it was still happening up to like 2010 in California prisons. Immigration laws restricted entry of populations deemed to be genetically inferior. Those are still happening. And Nazi Germany actually made laws justifying mass killings or genocide of individuals believed to be genetically inferior, the Jewish people. And those laws were modeled on the sterilization laws and eugenics movement in our country. So Hitler got those ideas from us. Your discussion posts, there's a lot more on this because it's interesting and it's very relevant to what's happening in our country right now. Next. A few more definitions. Genomics is a study of organization, evolution, and the function of genomes. Genomes is a set of DNA sequences carried by an individual. So you each individually have your own genome within your cells. Genes associated with hundreds of genetic diseases have been cloned and are being used to develop tests and new therapies. Like I said, certain cancers run in families. So they can clone that gene and just work with that gene and see if we can fix that gene. Diabetes runs in families. Let's see if we can just fix that diabetic gene. Genome screening can be used to assess genetic risk in families. Some diseases and disorders are more likely to be carried on than others. And then stem cell research offers hope for treating many diseases. Like I said, the genetic modification of food. How do we make it healthier? How do we make it last longer? Those are all genes that are inserted into our food. And now it's called genetically modified. The Human Genome Project was the international collaborative research program whose goal was to complete the mapping and understanding of all the genes of human beings. All of our genes together are known as our genome. And we have 3 billion base pairs in each one of our genomes. We'll talk more about this picture in a minute. The simplest type of variation in a genome sequence is called a single nucleotide change. It's officially called a single nucleotide polymorphism, or a SNP. So if you see person 1 has a C right here, but 2, 3, and 4 have Gs, that's a single nucleotide change. They also, person 3 has a C in this spot, and person 4 has a T. It's just one single nucleotide change. And scientists all over the world can analyze groups of SNPs to study differences among individuals at the molecular level. So whatever gene this is, say this is the gene for cancer. And this person with a C right here might be more likely to develop cancer as opposed to the people who have Gs right here. That's what they're studying. That was the point of the Human Genome Project, was to line up our entire 3 billion base pair DNA sequence and compare it to other people's and see, this is the gene for cancer, this is the gene for diabetes, this is the gene for schizophrenia, whatever. Bipolar runs in families. A genetic technology called a DNA microarray can be used to determine genetic disorders an individual has or will develop. They stick it into a computer and they can sequence the base pairs in a certain sequence fairly quickly. It doesn't take years like the Human Genome Project did anymore. In vitro fertilization is another technology that's come from all this. So when a couple can't conceive through traditional means, they need to look at other options. And I forgot to tell you that these are links. So if you click on them, it'll take you to a video explaining more about in vitro fertilization. The Human Genome Project had a link too. So I'll go through some of it. If you guys want to know more, I'll put links in there. So in vitro fertilization, when couples can't conceive, fertilization happens outside the body, followed by an intrauterine or into the uterus embryo transfer. So the uterus is the organ that babies are held in when a woman is pregnant, in case anybody doesn't know. It's called a uterus. Babies are not held in your stomach or in their stomachs. So intrauterine embryo transfer means they fertilize, they put the egg and the sperm together outside in a petri dish, and then they'll insert it into the woman. Genetic testing can be done at this embryonic stage before the intrauterine transfer to determine whether the embryo has inherited any defective genes. Animal cloning is another technology they use. Dolly the sheep was a breakthrough in animal cloning methodology. She was the first mammal ever cloned. And now they've cloned quite a few mammals. And a lot of it is to save some of these animals from extinction. If it's an endangered species, you clone it. There's problems, there's issues with that, but if we don't want that species or that animal to go away completely, you got to do something. So they learned how to clone animals. The choices we make with completion of the Human Genome Project, new methods of genetic testing, and the ability to transfer genes from one species to another, we're faced with many personal and social decisions. The ethical use of genetic information and biotechnology requires education and participation by a broad cross-section of society. The ethical use of genetic information. You see all over those ancestry tests, those 23andMe tests. If you've ever taken those or you know anybody who's ever taken those companies now have your DNA. What are they going to do with it? That's a question that has to be answered if you're going to run a DNA company. We can make informed personal decisions and formulate public policy only if we have knowledge of genetics and how genetics is used in biotechnology. There's laws now, they want to make laws about GMO foods.